Give a couple more minutes to see if anybody else wants to join us. Otherwise, I'll get started. But yeah, thank you for everybody who's joined us so far. Okay, I think that gives enough time and got a bit of material to cover, so I'll get started now. Just want to say hi, my name's Andrew Nicklawsky. I work as a biostatistician for the Cancer Center, specifically in the Biostatistics and Bioinformatics Shared Resource. As some of you may know, a large part of what we do is support clinical trial design for oncology research, especially for phase 1 studies. So the motivating factor behind these lecture series is fairly recent developments in trial design have led to a plethora of options that have been proven to perform better than the standard design, which most investigators are familiar with. Recognizing the NIH has called for use of these designs, and this talk is going to be the first in a series of lectures that we've kind of put together to help bring awareness to these new novel designs, how they work, and to encourage their use throughout campus oncology research. So like I said, this first lecture in our series will cover a little bit of background on phase 1 designs for those who aren't familiar with kind of the structure. The standard 3+3 approach, its deficiencies, and what other options there are available. Subsequent lectures will cover these designs in more detail. So as I mentioned, this talk is aimed at convincing you the 3+3 design is not ideal. However, if you've used this design, don't feel bad. It's really been a staple of phase 1 research for a long time, and it's really well known. So hopefully this talk will help you convince you to consider the alternatives in the future. So I guess I started a little early, but that's okay. It's always hard to tell with these Zoom meetings. But anyways, so and talking about clinical trial phases, many of you are probably already familiar with this, but just really briefly, trial design is typically divided into 4 to 5 phases, depending on whether you consider what account. A phase 0 trial is usually the first clinical trial done among people. These aim to learn how a drug is processed in the body, how it affects the body. And in these trials, we're generally looking at about 10 to 15 patients and giving them a very small dose of drug. Phase 1 trials, which is what we're focused on today, aim to find the best dose of a new drug with the fewest side effects. So we're going to look to test this drug in about 15 to 30 patients. At this point, we know the drug may help people, but we're not sure how much. That's not the focus of a phase 1 trial. Phase 1 trials are to test drug safety. So that's it. And if the drug is found safe enough, then we can proceed on to a phase 2 clinical trial. Phase 2 clinical trials is where we're further assessing the safety that we're looking at in phase 1, but now we're also considering if the drug itself works. So we're going to test that specifically usually amongst people with a certain type of cancer. These phase 2 trials are going to be done in larger groups of patients compared to phase 1 trials. We're going to be monitoring these patients to see if the drug works. However, at this stage, we're not really looking to see how it works relative to standard of care. We're just trying to measure the efficacy of the drug itself. If it is found to work, then we're going to move on to a phase 3 clinical trial, which is probably what a lot of people are familiar with. Phase 3 trials are comparing this drug to the standard of care drugs. These trials assess the side effects of each drug, which drug works better. They usually enroll around 100 patients or more. Often these trials are randomized, and we're looking at possibly more than 2 treatment groups because we're considering all types of care. Phase 3 trials are often needed before the FDA approve the use of a new drug for the general public. And then finally, we have phase 4 clinical trials, which are to test new drugs approved by the FDA. The drug is being tested now in several hundreds or even thousands of patients, which allows for better research on short-lasting side effects and safety. For instance, we might see some rare side effects that may only be found in large groups of people. So, like I said, this lecture series is going to be focusing on phase 1 clinical trials. So we're looking to see what the best dose is of a new drug with the fewest side effects, with the primary purpose being established safety. Once again, the endpoints are going to be safety, pharmacokinetics, and the toxicity profile of the drug. And like I said, the real important thing here is that while the drug may help patients, the efficacy itself isn't assessed until a phase 2 trial. So all we're really considering is the safety of this drug at this point. So when we talk about phase 1 oncology trials, the issue is they're often coupled with a very limited number of available patients. So this is especially the case when we're talking about rare diseases, which often happens with cancer research. So it's really important for us to use the resources available to us wisely. So we need to make sure we're designing our phase 1 study using an approach that's not only safe for the patients themselves, but also using all the information that we can from these very limited resources. When we're designing a phase 1 trial, there's a few different critical operating characteristics that we're interested in taking into account, such as the probability of each dose being the maximum tolerated dose, which I'll get into in a second, the expected number of patients treated at each dose level, the expected probability of dose-limiting toxicities, with the toxicity typically being defined using the CTCAE grade of 3 or 4, if you're familiar with that, with a duration that's too long as to prohibit retreatment, our probability of stopping early during the trial itself, and then the expected total number of patients. So what's going to happen is once we design the trial, our goal itself is to determine what the safety profile of the drug is, and then also identify a recommended phase 2 dose level. So historically, and what a lot of people are probably familiar with is the use of the 3+3 design. It's been a recent publication in, I believe, 2007 was found that approximately 98.4% of published phase 1 clinical trials rely on these 3+3 or other algorithmic designs, including the rolling 6 and accelerated titration. So the vast majority of trials, at least within the last 10 years or so, we're relying on these designs. And the reason is the 3+3 is very simple to use. It's very simple to implement, and the analysis itself is very simple. It's rule-based. There's no statistical models. That's very transparent, and it's societally accepted. Obviously, with 98% of trials using that. Now, briefly to touch on how the 3+3 works. Traditionally, when we're talking about cancer drugs, we're looking at cytotoxic agents such as chemotherapies. So what this means is that typically a higher dose level usually leads to a higher toxicity but also a higher efficacy. So therefore, what could happen is a dose level that's too low might be very well tolerated, not very toxic, but it's unable to induce sufficient efficacy. While a dose level that is too high can be very potent and efficacious, but very toxic. So patients can't tolerate it. So what we're trying to do is find a drug that is efficacious, but not too toxic. So with the 3+3 design, or a lot of designs, what we're trying to do is start at a very low dose and gradually increase these dose levels so we can find this maximum tolerated dose, the MTD, hoping that the MTD itself is the best dose for patients that is sufficiently efficacious without having too much excessive toxicity. A typical phase 1 3+3 trial is illustrated in this figure I have here, which shows 4 ascending doses on a vertical axis, which are then assigned to patients represented by circles sequentially and adaptively. Just something to point out, patients aren't really randomized in phase 1 trial because they're all receiving the same drug. So instead, what we're doing is treating cohorts of 3 patients specific for the 3+3 design at different dose levels. So what we're doing in this figure is having 4 different dose levels and escalating through that. So typically the rules of escalation for a 3+3 design follow these principles. If there's no DLTs, so 0 of 3 for the patients, we're going to escalate to the next dosing level. If there's a DLT amongst those 3 patients, we're going to stay at that dosing level and have an expansion cohort where we assign 3 more patients to that dosing level. If there's no additional DLTs, so only a maximum of 1 of 6 at that dosing level, we're going to escalate. If there's 2 or more, though, we're going to stop and we're going to say the prior dose level is our MTD. Now, this is the same kind of principle applies if we're just looking at 3 patients. If there's 2 of 3, we're going to stop and the MTD is the prior dose level as well. So, for example, in this figure, what happened is we enrolled 3 patients at the first dose level, none of which experienced a DLT. So we escalated to the second dosing level. Once again, none of these 3 patients had a DLT as well. So we went to the third level. At this point, the first cohort of 3 one of them had a DLT represented by that red circle. So we enrolled an expansion cohort of 3 additional patients staying at the same dose level, of which none of those experienced a DLT. So by our logic, then we would escalate to this final 4th level, where unfortunately 2 patients experienced a DLT, meaning that we're going to halt the trial and say that level 3 is our MTD. So in essence, what we're saying is the MTD is the highest dose level with 1 or 0 basically DLTs out of our 6 patients, which means that our targeted toxicity probability at the MTD is going to end up in the range of 17 to 33%, where 17 is 1 out of 6 DLTs to 33% being 1 out of 3. So given how simple this design is and its implementation, why should we question it? Well, there's a couple of different things to take into account. First being a recently published paper has found that at least, I believe it was published in 2010, 25% of oncology agents registered by the FDA are labeled at dose different from what is identified in phase 1 trials, which means that our phase 1 trials are not really actually doing a good job in identifying the correct dose. Now, that might have a little bit to do with efficacy and so on, but really the purpose of our phase 1 trial is to find the correct dose. So it's not good if 1 out of 4 times it's not doing that. And drug development itself is really kind of in need of innovation. It's very expensive. A recent survey published in 2016 found that the average out-of-pocket cost per approved new drug in the US is about $2.5 billion, which is huge. These drug development itself is really slow, and there's a high failure rate. So what does this have to do with the 3+3 though? Well, we have numerous publications. Here's a select 3 that have shown that the 3+3 is very inferior in many ways. So some of these disadvantages are the 3+3 design utilizes a fixed cohort design. So we can't treat more than 2 cohorts of 3 patients at a given dose. By nature, we're just looking at 6 patients max at a dose level. When we're looking at for the MTD, it's going to be determined within a range with no pre-specified toxicity probability. That means that if I'm a clinician and I think that it's okay if my drug has 25% toxicity probability, I can't do that because it's going to end up somewhere between 17 and 33%. It's going to be 1 out of 6 or 1 out of 3 patients max having a DLT with 3+3 design. And with that, without having a pre-specified toxicity, then there's a risk of causing a severe toxicity far different to what we deem acceptable, because we don't really have a lot of input then on what that should be. There's also a lower probability of selecting the true MTD, which has been proven in these papers. Which can be a problem, because if the MTD is underestimated, patients can be treated then at a subtherapeutic dose level in the phase 2 trial. There's also a higher variability in the MTD estimates because we're only utilizing data at the current dose to choose the next dose, which can cause uncertainty around the estimated DLT risks at each dose. Other issues are we can't use the 3+3 for delayed toxicity studies, which often occurs. And the performance is very dependent on the number of doses that we decide to use initially. So given that there's all these kind of bad things about the 3+3, what other options are there available out there? Well, looking at the timeline of publications. So in 1990, in order to overcome these deficiencies that were known for the 3+3 design, a model-based approach called the continuous assessment method, or the CRM, was developed. However, as you can see, there's a pretty large gap before additional designs are, and that's because this design met a lot of resistance. It was unfamiliar. There's a lot of assumptions that need to be made in terms of the shape of the dose toxicity curve. It's statistically complex. You needed specialized software to perform it. There had to be then a lot of increased communication between PIs and the biostatistician to actually design and implement it, because it's very intensive design. In response to that, recently, since 2007, there's a new class of designs created called model-assisted designs, such as the TPI, the MTPI, the Boyne, and keyboard design. So I'm going to go into a little more detail on the two differences between those, but basically there's these 2 different classes now of designs in addition to algorithmic designs, which are model-based or model-assisted designs. And like I said, I'm going to go into a little detail covering some of the specifics of the designs, but the next lectures in this series are going to go into a lot more detail than what I'm going to give. So if there's any confusion or a little ambiguity about the details, that's okay, because we're gonna be talking about them a lot more in the next few lectures. But first, just a brief overview of model-based designs. So the great things about model-based designs is they account for that variability in the MTD that we have concerns about with the 3+3. They're very flexible and they can be very powerful if there's the right the correctly specified dose-limiting toxicity curve. The most well-known design of this class is the continual reassessment method, or the CRM. So basically what we do with this is we begin with a priorly specified dose toxicity curve, and we continuously update this curve based on the accruing outcomes during the trial. So it's adaptively changing as we go along. So as we progress through the trial, patients are then assigned to the dose that corresponds to the estimated DLT probably closest to the pre-specified target that we've done before we even initiated the trial. And then as we resolve the trial, these DLT probabilities are then being derived from this updated dose toxicity curve. There's a lot of extensions to this design, such as the EWOC, the time to event CRM, the Bayesian model averaging CRM. But basically, compared to the algorithmic designs, like the 3+3, these really have superior operating characteristics. And here are some of the advantages. So when we're talking about targeting a specific DLT rate, like I said, with rule-based designs, it's pretty unclear because that's a wide window. With model-based designs, we can clearly define it and it's flexibly chosen. Like I mentioned before, with the goal of an early phase dose escalation study with a cytotoxic oncology drug is to estimate the maximum tolerated dose. So we're looking for the highest dose that does not cause unacceptable side effects. Statistically speaking, we're looking for the dose where the probability of a dose-limiting toxicity is equal to some pre-specified target level, which typically falls somewhere between 0.25 and 0.33. Using the model-based designs, the MTD at the end of the trial then is going to relate to the targeted probability of the DLT that we pre-specified before the trial even began. However, and I said this is not the case with rule-based designs, since there's that large window, and it can vary greatly. Another great characteristic about model-based designs is we're going to see a lot more patients being treated at the optimal dose versus very few being treated at the MTD in a 3+3 design. Then on top of that, then we'll also see a lot of people treated at subtherapeutic doses then for a 3+3 design versus hopefully relatively few with a model-based approach. Now, a big thing that's an advantage for these model-based design is we're really making efficient utilization of our available data. Model-based designs allow the estimation of the MTD together with informative measure precision. So under a 3+3 design, there's a lot of uncertainty around the MTD selected at the end of the trial. When we're thinking about our 95% confidence intervals for the probability of a DLT, it's either going to be somewhere between 0.004 to 0.64 for 1 out of 6 DLTs, or 0 to 0.71 when we're considering 0 of 3 DLTs. So that's a pretty big window, and that's not too great. The other big advantage of model-based designs is it's pretty smooth and straightforward to extend them to more complex questions. Although there's some extensions for rule-based designs to deal with drug combinations, they suffer from the same drawbacks as like a simple 3+3 design. Model-based approaches are very flexible, and they supersede rule-based designs for more complex questions, like when we're assessing drug combinations or toxicity together with efficacy. So where the goal of a dose escalation study is to understand a dose-response relationship, only the model-based approach will be expedited, whereas the 3+3 designs do not really adequately characterize the dose-response curve and therefore are kind of useless. The other big thing is if we need to deviate from our plan, like adding other doses, looking at different number of patients on doses, it's really hard and near impossible to do that with 3+3 design. Whereas with model-based approaches, this usually can be easily accommodated. So with all these positives I just talked about, why aren't everybody using the CRM or a model-based approach? Well, like I said before, it's for the reason that they weren't adopted initially. They're pretty statistically complex, and it's not really easy to explain what's happening in terms of your decision making. What you're doing is you're repeatedly model fitting and estimating during the trial, which can be pretty intensive and not very efficient. Therefore, it's viewed as conceptually and computationally complex and really kind of a black box, because usually you have your biostatistician or something doing something on his computer, and it's not really clear why decisions are being made. So in response to that, there's a second class of designs being made, which are interval or model assisted designs. What these are meant to do is harness the power of a model-based design, not have those kind of bad issues that I just talked about. So the major benefit of these designs is, like algorithmic-based designs, the dose escalation and de-escalation rules can be tabulated before the trial even begins. So unlike a model-based design like the CRM, which assumes a dose toxicity curve across all the doses, the model-assisted designs often model only local data, usually using a binomial model, thus rendering it possible to enumerate the dose escalation and de-escalation rules before the trials begin. Some examples of these model-assisted designs include the modified toxicity probability interval, which is the MTPI, its variations, the MTPI2, the Bayesian optimal interval design called the BOIN, the keyboard design, and then a bunch of other combinations thereafter. So there's been a lot of papers written about their superior performance 3+3 designs, specifically in their accuracy of identification of the MTB, the allocation of patients to the MTD, and then just the risk of overdosing patients. And like before, I'm just going to very briefly talk about 3 of these designs. Subsequent lectures in this series are going to go in a lot more detail than what I'm covering here. But basically, this image shows what the MTPI, the keyboard, and the point design kind of use to guide their decision rules. So the MTPI is figure A, the keyboard design is figure B, and then figure C is the BOIN. Excuse me. So starting with the MTPI design, what we're going to do is we're going to define 3 toxicity probability intervals, an underdosing interval, a proper dosing interval, and an overdosing interval. So for example, in this figure, our underdosing interval is going to range from 0 to approximately 0.25. Our proper dosing interval is 0.25 to 0.33 probably, whereas overdosing is 0.33 to 1 respectively. So what we're going to do is, given the data that we observed at our current dose, the MTPI is going to make the decision of dose escalation and de-escalation on the base of a unit probability mass, or UPM, of those 3 intervals. So say we denote our true DLT probability being P. The UPM of these intervals is then defined as the posterior probability, so the probability calculated after looking at the data, that P resides within that interval divided by the length of the interval itself. So if that UPM then is found to be the largest one, would be like underdosing, then we're going to escalate to a higher dose level. Otherwise, if we're finding it to be probably going to lie within the proper dosing, will stay. Overdosing will de-escalate. So that's just very briefly how that design works. Now, a problem that you might already see, which we'll go into more detail in a subsequent lecture, is those intervals kind of are dependent on the actual size itself, which is what the keyboard design then tries to address in figure B. So similar principle, except now we're going to make a bunch of different equal lift dosing intervals to represent the potential location of our probability of having a dose-limiting toxicity. Then we're going to define this, what's called the target key, which is what we think our proper dosing interval is. So how this design works, and similarly to the MTPI, we're going to look at the relative position between the target key and the strongest key, where the strongest key is whichever one has that largest UPM, and make a decision whether to escalate or de-escalate if that strongest key is larger or smaller, or on the scale relative to the target key. So, for instance, if the strongest key is on the left of the target key, that would indicate that we're probably underdosing, so we need to escalate. Then finally, pretty simply, the Boyne design makes dose escalation, de-escalation recommendations simply by comparing our estimated DLT rate with the pre-specified dose escalation and de-escalation boundaries. So as you can see in this figure, we have the lower boundary and the higher boundary, and then just a small interval for where we stay. So basically what we're going to do is treat the first cohort of patients at the lowest dose, or whatever the clinician specified as the starting dose. We're going to assign the dose to the next cohort of patients on the basis of whether that calculated DLT rate is less than the boundary pre-specified or greater than. And then if it's falling obviously in between them, we're going to stay. So we'll just keep repeating that until we meet the pre-specified maximum sample size for that trial, or until we reach a specific number of patients treated at a single dose level. Often this is 12. And then once the trial ends, we're going to select the MTD as a dose in which the DLT estimate is closest to our target. As I mentioned before, the really nice thing about these designs is their ability to have pre-tabulated decision tables. And for instance, this is what one of those would look like if we're using the Boyne design. So in this case, I specified having 5 doses. We're going to treat cohorts of size 3, and we're going to treat 10 cohorts. So already we can already say we're going to treat 30 patients off the bat, which isn't necessarily something that we can do with the 3+3 trial. We're going to stop the trial if 12 patients are deemed are dosed at a certain level, and we're shooting for a target toxicity of 0.25. So we can already say what we want our toxicity rate. And then the other nice thing is with this design, we're going to add a dose elimination criteria of 95%, which means that if the probability of a dose exceeding the target toxicity level that we had specified exceeds 95%, we're just going to eliminate that dose entirely. So like I said, just briefly looking at this figure, you can see we can make our decision based off the number of patients, which is at the very top, and then determine if we're going to escalate, de-escalate, or eliminate that dose level based off of the responses that we're seeing. Very easy to use, and you don't need input. You don't need someone to be fitting a model. You don't need to do any of that. And similar to the 3+3 design. So one last thing, as I mentioned, with the 3+3 design, it's really hard to deal with late onset toxicity, which is actually pretty common in our targeted therapies and immunotherapies. It's logistically very difficult to handle because we can't make a decision in like the 3+3 design until we've observed a DLT. So that can really lead to a long trial, which is not ideal. So for example, if we have a DLT that takes up to 8 weeks to evaluate and our accrual rate is 1 patient per week, And on average, we're probably going to get about 5 new patients that could have been accrued for the trial while we're just waiting to evaluate the 3 patients' outcomes, which is a waste of resources, and it's not ideal. So the question then becomes, how can we treat these new patients with our previous outcomes being pending? To deal with that, there's some model-assisted designs, such as the tight Boyne or the tight keyboard. There's also the model-based designs, like the tight CRM, where tight just means time to event. And we're going to have a lecture on this, I believe lecture 5, talking a little bit more about how to deal with this. So finally, I've mentioned that we have all these designs and they perform better, but I haven't really pointed to any proof other than just conjecture. So here's a sample publication kind of giving some proof on why these designs perform better than the 3+3. What this paper did is they compared a bunch of different model-based designs, such as CRM, as well as model-assisted designs like the Boyne, the MTPI, and keyboard with the 3+3 trial. And the results are really interesting and give some interesting insight into these different designs' performance. So what this trial did was, or what this study did, is run a bunch of different simulations and then look at different metrics that they're interested in, such as accuracy, safety, and then reliability. So here we have 4 figures dealing with the first 2 of those metrics, accuracy and safety. So in figure A, they're looking at the percentage of correct selection, which they defined as the percentage of simulated trials in which the target dose was correctly selected as the MTD. And then in the case when the dose levels are all above the MTD, the percentage correct selection was defined as the percentage of early termination of the trials. Figure B is going to show the average percent of patients who are assigned to the MTD across simulated trials. When all dose levels are above the MTD, they're going to use the average percentage of patients not enrolled into the trial for this metric. So as you notice, the 3+3 is absent because they're comparing all these different designs on the x-axis versus the 3+3. So when we're looking at figures A and B, larger values, positive, are going to indicate better performance than the 3+3. And some things just stick out immediately. In terms of accuracy of correctly selecting the MTD, we see that the CRM, the MTPI, the Boyne and keyboard designs are pretty comparable in figure A. And they also substantially outperform the 3+3 design, keeping in mind that for instance, for the PCS, percentage correct selection, that's taking the simulated results, CRM, Boyne, whatever, and subtracting off that of the 3+3. So for the CRM or the Boyne, we can see on average, that's almost 20%, which is pretty significant. In terms of percentage of patients treated at the MTD, the MTPI and CRM design allocate slightly more patients than the Boyne and keyboard design, but we can also see, based off these box plots, that the latter are less variable. So now looking at safety, this study looked at 2 different metrics here. One being in figure C, the percentage of simulated trials in which a toxic dose was selected with the true DLT probably greater than 33% being selected as the MTD, which means that we're selecting too toxic of a dose. Not great. Figure D shows the average percentage of patients assigned to the toxic dose with a true DLT probability of greater than 33%. Now, in this case, we want smaller values relative to the 3+3, which, as you can see on both, unfortunately, we have slightly larger for like the CRM, the Boyne, the MTPI, the keyboard, but I wouldn't say it's too much. And the reason being is these designs are a little more aggressive than the 3+3, other than the BLRM and the EWOC in terms of having patients or selecting doses that are a little too toxic, but it's marginal. And as we'll see actually in this next slide, this isn't the entire picture. So the second half of that is looking at reliability, which are A and B of these figures. So they looked at the risk in figure A, the risk of overdosing, which is defined as the percentage of simulated trials with more than 50% being treated at a dose above the MTD. That is, we're measuring the likelihood of a design assigning more than half the patient's doses above the MTD. And then in figure B, the risk of poor allocation, which was defined as a percent of simulated trials in which fewer than 6 patients are treated at the MTD. So as we can see by these results, in terms of the risk of overdosing 50% or more patients, lower is going to be better. And the Boyne keyboard designs perform the best. And the CRM and MTPI perform comparably to the 3+3. Which, so taken in at having a little more toxic dose sometimes than the 3+3, you're not overdosing more patients than the 3+3, and which is great. So you have to take this into consideration. And then the other kind of big thing here that they looked at in terms of reliability is this figure C, which is the risk of irrational dose assignment, which they defined as a percentage of time that the design failed to de-escalate dose when greater than 2/3 or half, 3 out of 6 patients had DLTs at a dose. Now, the really striking thing here is the model-assisted designs outperform the model-based designs substantially. They didn't do that. The model-based designs have an 8 to 55% chance of failing to de-escalate the dose when 2/3 of or greater or than or equal to 3/6 patients have DLTs. That just never occurs in the MTI, PI, the Boyne, the keyboard. And this is most likely due to these model-based designs relying on assumed model to make decisions for dose assignments, because that can cut both ways. That can make it really efficient, but if you misspecify your curve, these estimates can be really biased, causing irrational dose assignments. This isn't the case, though, amongst these model-assisted designs, as they don't have any model assumptions and they impose restrictions that would not allow this behavior to even occur to begin with. So just with this one paper, we see already overall, compared with the 3+3 design, most of these novel designs yield better accuracy for identifying the MTD, and they allocate more patients to the MTD. In particular, the Boyne design and keyboard designs are really attractive because they perform virtually the same in each metric and are really transparent and easy to implement. So briefly, in conclusion, there's a lot of different designs available other than the 3+3. I only presented one paper, but there's a significant amount of research that demonstrates them outperforming the 3+3, and especially with the advent of model-assisted designs. The major barrier of transparency in these has been mostly eliminated because you have these pre-tabulated tables that allow you to just make decisions. It's pretty easy to understand what's going on mechanically with these designs. So we really encourage the use of these novel designs and think you should consider them if you're looking to design a trial in the future. Just to give a little plug for our group, If you're looking to do an oncology, excuse me, phase 1 trial or any trial to be in, we're here to offer you any assistance you need in terms of your protocol development, help you prepare for the IIT committee, for PRMS, all that covered by the Cancer Center. So it's free advice. We're going to help you design your trial. And then obviously on top of that, we offer services to help you with your grant proposals, other analysis, and so on. So next week, my colleague Elise is going to talk specifically about the Boyne design. It's going to be at the same time on Thursday, and it should be a really great talk. After that, there's going to be 3 more lectures covering the MTPI and keyboard design, as well as the CRM. And then finally, we're going to end talking about delayed toxicity. Now, I know we don't have a ton of time left, and I probably moved through things a little too quickly, but is there any questions that anybody had?
Great talk. I just had a quick question. My microphone was muted there. So you talked about phase 1s, but is it possible to use the same method in, say, like a phase 2 or phase 3 for safety issues, kind of unrelated to efficacy, just for determining stopping rules?
Yeah, I wouldn't say, these specific designs are related to phase 1 trials, but there are adaptive approaches that can harness some of what you're talking about. So they do exist. I wouldn't necessarily say that like the Boyne it is a phase 2 trial design, but there are phase 1, phase 2 hybrids that utilize. So I think I'm kind of answering. It's not these designs specifically, but there are designs out there that can do what you asked. All right. Well, if there's not any other questions, I'll just end by saying I think that if you want to access this talk, I have a bunch of references still. I can email it to you. I can send you some of these papers. Feel free to reach out if you have any questions. I believe this will be posted to our YouTube page if you want to watch it later. But otherwise, I'll hopefully we'll see you next week then. And thank you for joining us today.